Okay, so good afternoon, good morning, but especially good evening today, because the speakers of today, Shubha Tole, is actually in her late evening in India. And so it's really my pleasure, my great pleasure today to introduce Shubha for this fantastic series of seminars. So the Worldwide Neurodevelopmental Series organized by Denis. And so I will start by saying a few words about her career. So actually, Shubha is from India, and she studied also in India, in Mumbai, and she studied life science and biochemistry. And then very early in her career already, she moved for her master and PhD to California, to Caltech. And in the Patterson lab already, she started falling in love with something that she actually continued to work on since many years, namely the development of the brain, and in particular the patterning of the brain. So after her PhD, then she decided to move to the University of Chicago in Elizabeth Grove lab for a postdoc. And there also she actually got really one of the experts in brain development and patterning, as she continued to study there this specific part of the brain development. And then after that, she actually returned back home, and since then is in the Tata Institute in Mumbai, in India, where she joined at that time as a faculty member, and now obviously she gained more senior position there, and she is now actually always also the dean of the graduate studies. So a few years ago, she also decided she was not done with moving, and she came to Europe for a sabbatic. Actually, she joined the lab of Denise Boudon in Geneva. And I think she also used that time to visit a lot of labs in Europe, because actually it's in this way that I got to know her. This was back in 2014, when I was still a postdoc at the Helmholtz Center in the lab of Magdalena Goetz. And I have to say, I was not only fascinated by her exciting science, but also at that time I had really the chance to get to know another side of Shubha, which is her dedication in following, encouraging young scientists, and especially female scientists. She's really caring about this part of science, and she's actively working on mentoring, and she indeed has a live blog, and she's doing a lot of seminars to advise young people in their career by sharing her experience, which I think is a very important way of mentoring young people. So back to science. At that time, obviously in this time she got a lot of grants, awards, and prizes, and I would just like to mention a few of them. So she got the prize for life science. She got I'm not sure I will pronounce correctly, the Bhatnagar Award, which is one of the highest scientific honor in India. She also got a research award for innovation in neuroscience by the Society of Neuroscience, and many, many more. So what you can imagine already is that the main interest of Shubha's lab is very broad, and she wants to understand how the brain develops, but specifically she's focusing on trying to understand molecular mechanisms and signaling pathways, which are involved in cell fate specification and patterning. And today in particular, she will tell us about hippocampus and how to make an hippocampus. So thank you very much for being here today, and I'm really looking forward to your talk. Please share your screen.
Thank you so very much, Sylvia. Happy memories of our first visit about 6 years ago. And it's wonderful to catch up. And thank you so much to Jenny for inviting me to speak here. And indeed, for creating such a wonderful forum to integrate people across the whole world, no matter what time zone. I mean, this is something really special. So it's a wonderful contribution to neuroscience. And I'm happy to share our story. The hippocampal work that my team of wonderful students and postdocs have been working on for now 2 decades since I started my lab in Mumbai. My God, 2 decades, okay. All right, it's flown. I need a new hippocampus now because my memory doesn't serve as well as it should. Let's build one in the mouse for now. Let's see, share screen.
I can tell you when everything is shared and I can see everything, then I will mute myself.
Is it shared? Not yet.
Perfect.
Okay. Slideshow? Yes?
It's a bit cut, but yes. I just see half of your screen, actually.
Oh. It worked perfectly well when we were testing, huh?
Yeah. It's just, I just see half of it.
Now?
Perfect. Okay.
Okay, all right, let's go. So, how to make a hippocampus? This is our beautiful campus at the Tata Institute of Fundamental Research in Mumbai. Over here is the Arabian Sea. And this is the view from our building, this lovely long walk. People stroll here in the evening, watch the lovely sunset over the Arabian Sea. Students sit here, gazing into the sunset, wondering when they will ever graduate. The usual. It's perhaps just a little warmer here than in some parts of the country, but the thoughts and sentiments much the same. So, how to make a hippocampus? I'm going to begin the way our journey into this wonderful structure, the hippocampus, began. And no, we don't all have hippocampal tattoos. That was just a find of Denise, but now maybe we should consider getting them. So, okay, here is the mouse brain, our model system of choice. I don't need to introduce to this audience that this is a mid-gestation mouse brain, about embryonic 12.5. And some time ago now, we began focusing on this transcription factor, LHX2, a limb homodomain transcription factor, simply because it caught my interest when I was a late-stage postdoc in the Grove Lab. And the reason it caught my interest was because it was expressed very nicely in the dorsal telencephalon. Here is an in situ. And it appeared to exclude almost completely this small structure at the end of the telencephalon, the cortical hem. The cortical hem, Wnt and BMP-rich signaling center, was something that we thought would be important as a patterning center for the medial telencephalon. But at the time, we didn't know what it did. And this transcription factor sort of led us there. Here then is a false color double in situ, an overlay of Wnt2b expression in the cortical hem, and DBX1 expression here on the other side of the strip of cortical neuroepithelium, a structure which the Grove and Ragsdale labs named the antihem. And the idea was that the hem and the antihem could be two signaling centers that sort of flank the expanse of the cortex, both the neocortex and the hippocampus being cortical primordium. Well, so we looked at the LHX2 null mutant, which we obtained from Denny Porter at the NIH when he was a postdoc with Heino Westphal, because he had created this mutant, reported that it died mid-gestation because of a hematopoietic defect, and reported a shrinkage of the dorsal telencephalon. With Nissl stain, it was not possible to say what that was. And when we looked, we found, amazingly enough, that the dorsal telencephalon contained no cortical primordium. It seemed to be entirely hem and antihem. Okay, so it seemed to us, so to skim over work, we know that this is not due to over-proliferation of the hem and cell death of the tissue here. We were able to discover that the cortex effectively forms by the presence of LHX2 suppressing these two fates, hem and antihem, that the presence of LHX2 in these structures sort of prevents them from becoming hem and antihem, and in that sense permits cortical fate. And we showed this by doing a mosaic experiment. Okay, a mosaic experiment, which I'm just going to summarize here since it was published some time ago. But we created embryonic stem cell chimeras. which were GFP expressing and null for LHX2, and mixed them with wild type morulae, and then implanted them into pseudopregnant mice and so on. Of course, we did this with control embryonic stem cells that were not null for LHX2. And in so doing, we obtained embryos that had clusters, clumps of cells. I won't call them clones since we do not know that they were clonal or not, but clumps of cells, which amazingly enough, recapitulated what we see in this null mutant. Here in the null mutant, everybody is mutant for LHX2. And here, only these colored clumps are mutant, and they're embedded in wild type tissue. And we were able to show pretty much cell autonomously that LHX2 is required to retain cortical fate. And in the absence of this, of LHX2, clumps that are medial take on hem fate, and clumps that are lateral take on anti-hem fate. So this, in some sense, sets up what I'm going to allude to during my talk, that LHX2 has spatially distinct functions. Okay, it suppresses two different fates in two different parts of the telencephalon. And I'm going to show you a little bit of this data, not as much to make the point about cell fate, but to say that this experiment, while we started it trying to understand what LHX2 does, and okay, it suppresses these fates, we created a transplant experiment that I had long been wanting to do. I had wanted to, much like the Shubha and Mangold transplant experiment where they transplanted an extra organizer into a host and then got two body axes. I had long since wanted to transplant the hem, in different places in the cortical primordium, but we couldn't do this experiment early enough in development to actually test it meaningfully and do it in utero. Now, this genetic perturbation, a chimera, a mosaic of null and wild type cells, created for us effectively ectopic extra hems embedded amongst wild type tissue. So we were able to ask, well, what do these ectopic hems do when they are juxtaposed to wild type tissue? Okay, so here is just one slide summarizing this data. Here are control chimeras where both green and not green cells are both wild type. And you can see that the patterning is not affected, that EMX1 expresses in the dorsal telencephalon as it should, and is nicely complementary to Wnt2b, which marks the hem. Now here are the chimeras with wild type and LHX2 null cells. And you can see that these clumps express in the medial part of the telencephalon, the clumps express hem marker, here LMX1A, they also express Wnt. And these are actually the same section, simply photographed in green and then processed for in situ. So I will overlay them here, and you can look at either this triangle or this big rectangular clump or this thin line, any of them. They don't have LHX2, they can't become cortex, they become hem, and only the medial ones do. What I won't show is that lateral clumps become anti-hem. Well, so these ectopic clumps, we found induced ectopic hippocampi. So here at embryonic day 15 is the dwindled normal hem. It shrinks with time and becomes, basically that's where the fimbria forms. And here is a dentate gyrus marker, PROX1. And you can see the baby dentate forming, the dentate niche here, the stream of cells migrating, forming the beginnings of the dentate. That's the control. In the chimera, we have many, many ectopic green mutant clumps, and each one becomes a hem. And next to each hem develops an independent PROX1 positive clump. And you can even see some sort of streaming going on here. And we looked with, so these chimeras don't make it to birth. So we cannot do experiments of animals with multiple hippocampi running around and do spatial memory tests. But a little older chimera, embryonic day 17, I'm just going to show you this because I continue to marvel at how it looks. Here is LHX9 labeling the wild type, the control, dentate, and CA3. Ignore this staining in the thalamus. And here is a chimera with an even more dwindled set of ectopic hems at E17. And each one has now induced an ectopic hippocampus. So in this hemisphere, we identified up to 5 independent hippocampi. They don't all look like C shapes because we don't have control over which orientation that it's going to form in. But the other hemisphere was a little more well-ordered, and you can see at least 2 nice little V-shaped dentates with a merged giant Ammon's horn. So kind of fascinating. So we were able to show then that the hem is an organizer for the hippocampus. So just summarizing this, these data because it sets up the rest of the talk. We found then that LHX2 suppresses hem fate in the medial telencephalon, and that LHX2 null clusters form hem, but only medially, and that the hem is an organizer for the hippocampus. So in some sense, the hippocampus forms where it forms in the caudomedial telencephalon because the hem forms where it forms. So in terms of how the brain is structured, we've pushed back the question one step of why the hippocampus is where it is. Which then begs the question, why is the hem where it is? Okay, what positions the hem? And why doesn't the hem form laterally? If we were to persuade the hem to form laterally, would we get a hippocampus there? I don't have the answer to that. A postdoc in my lab, Nidhi Saikadkar, is actually trying that experiment as we speak. But what I will share with you is the work of a talented postdoc, a former postdoc, Geeta Godbole, who basically found a way to extend the hem all over the telencephalon by looking at interactions between LHX2, PAX6, and FOXG1, three sort of fundamental patterning transcription factors in the brain. Okay, we obtained conditional mutants for all of these, LHX2 from Ed Manuki, the PAX6 conditional from David Price, and the FOXG1 from Goichi Miyoshi and Gord Fisher. So here's the expression pattern then of LHX2 and PAX6. They are broadly complementary. LHX2 is medial high, lateral low. PAX6 is lateral high, medial low. PAX6 is in fact critical for the formation of the antihem itself. In a PAX6 mutant, the hem forms where it does. I won't show you that data. There is no problem with the hem that we could find. Now, Geeta, see her here, funded by the Wellcome DBT Early Career Fellowship. She wanted to test whether this PAX6 presence laterally is part of what prevents the hem from forming there. Why does the hem restrict itself medially? So here's the normal hem. Here is the LHX2 conditional mutant hem. Here is it's on a Cre-ER background and tamoxifen given at embryonic 8.5. And you can see the hem extends as it normally would in a null mutant or a conditional which is induced at 8.5. And you can see that this stretch is not hem, it's basically anti-hem. Okay? And in the LHX2 PAX6 double conditional induced at 8.5, you can see that the hem extends farther to basically the paleo-subpaleo boundary. And in fact, this choppiness is very interesting because what she showed, so since this was a Cre-ER with tamoxifen, we would get occasionally a clump or two in which the PAX6 had not been completely floxed and which escaped the PAX6, the recombination. So these clumps that weren't able to become hem turned out to actually be PAX6 expressing. So in that sense, Geeta showed very nicely that PAX6 is what restricts the presence of PAX6 is what permits this tissue to become anti-hem, and in the absence of PAX6, this tissue also becomes hem. And here are some nice quantitations where this is the extent of the control hem normalized to the length of the dorsal telencephalon. And here in the LHX2 mutant, the hem expands a great deal. And in the LHX2 PAX6 double mutant, it expands farther still. So PAX6 then is one of the genes that prevents the hem from expanding all the way laterally. Geeta also looked at interaction between LHX2 and FOXG1. Both are expressed well in the dorsal telencephalon. And in fact, FOXG1 also expressed in the ventral telencephalon. Isang Lai's lab and Karina Hanashima showed very nicely that in the absence of FOXG1, the ventral telencephalon doesn't get specified. So in fact, you have an entirely dorsal structure. What Geeta did was looked at interaction between these two. But before that, she discovered in a nice collaboration with Bin Chen, who had done ChIP-seq for FOXG1, that FOXG1 basically occupies loci, potentially regulatory loci, three nice peaks on the LHX2 locus. Okay, so this is suggestive that FOXG1 actually regulates LHX2, and Geeta showed that indeed it did. So first, here is a luciferase assay for the same three peaks that FOXG1 occupies, and shows nice induction of the luciferase. And then Geeta looked at the double conditional, which, as I said, we obtained from Goichi Miyoshi and Gord Fisher. So here now is a Cre-ER This is a single FOXT1 conditional because she first wanted to test whether FOXT1 regulates LHX2. And here, induced with a low dose of tamoxifen at embryonic 8.5, she would get these vesicles which broadly are lacking FOXT1. And because they're lacking FOXT1, they display the FOXT1 mutant phenotype of lacking also the ventral telencephalon. But interestingly, because it's a low dose of tamoxifen, some cells appear to escape, and broadly, there are territories that are negative for LHX2 expression that we can detect, and territories that still continue to express LHX2. So in data that I won't show you because it's published already, Geeta found that by increasing dose of tamoxifen, she was able to effectively achieve a double null by simply giving a lot of tamoxifen so that the FOXT1 is completely recombined and the LHX2 then completely gets wiped out. So in some sense, she didn't need to actually do the double conditional because removing FOXT1 completely did the job. However, it's these low dose embryos that were very interesting to us. And the reason they were interesting is that, remember, basically LHX2, we showed, suppresses hem and anti-hem fate. And where there is no LHX2, there could be hem. And indeed, okay, Wnt3a expressing hem is nicely induced in these patches that are negative for LHX2. And what's very interesting, so these are serial sections, and what's very interesting is that the interspersed patches which do have LHX2 do not become hem. So the very strong sort of LHX2 yes, cortex, LHX2 no, hem is still maintained here. Now, I should also say that the FOXT1 null mutant not only misses the entire ventral telencephalon, it also misses the lateral cortical fates. It lacks the lateral cortical fates. So all of this is essentially medial tissue sort of stretched around into one giant vesicle. So which is why we're able to see hem everywhere. And now to ask, if this entire tissue now is either LHX2 positive tissue or ectopic hems everywhere, do each of them induce hippocampal markers? And indeed, lo and behold, they do. Every patch that is LHX2 positive expresses FP1, which is an early E12.5 hippocampal marker. So in so doing, Geeta was able to show in one fell swoop that removing FOXT1 is in some sense as effective as removing LHX2 in terms of creating ectopic hems, but you only get them where you also lose LHX2. So where the FOXT1 to LHX2 regulation is effective. And when you have patches that continue to express LHX2, there you get hippocampus. So this sets up an interesting question, which we then set out to explore. Okay, what we've shown so far is we have a hem. Okay, we've shown that the creation of this hem fate is nicely repressed by three molecules. It's repressed by LHX2 and by FOXT1 and by PAC6. And we also show a regulation of FOXT1 to LHX2. And what I didn't show here, but we've published, is LHX2 to PAC6. So it seems to be a nice little consortium and a nice tight way of keeping the hem where it is. A very important job because where the hem is, there the hippocampus forms, and you want the hippocampus to form in its right place. So it's important to restrict the organizer first. Okay, so now we have a hem. Yet, LHX2 expression persists in the neuroepithelium. That's not hem, not anti-hem. Okay, so here's hem, it's E12. LHX2 expression persists, E12, E15. Is the job of LHX2 limited to preventing these tissues from becoming hem? And once that job is done, that's that. Or does LHX2 have an additional job here? Because remember, what I was showing was you only get hippocampus where you have LHX2 expression. So is LHX2 part of the machinery to actually allow the responding cells to become hippocampus? In order to address that, well, we sort of stumbled into an answer to that, studying not LHX2 or PAC6 or FOXT1, but studying a completely different molecule called LDB. Okay, LDB, limb domain binding protein, binding factor. This molecule, LDB, has many names, and I'll just introduce them all to you. This is an E12.5 in situ, just to show where LHX2 is expressed and where LDB1 is. It was also called NL1 or CLIM, cofactor of limb. LDB1 is ubiquitous, it's everywhere. There is a partner LDB2 that's more restricted. And LHX2 and LDB have in fact been incredibly beautifully studied in the fly, where the LHX2 ortholog is apterous and LDB1 is chip. Okay, and this apterous and chip partnership has been elegantly studied, and I'm going to talk to you about it because we modeled our studies based on exactly that. Okay, LHX2 and LDB1 are known to bind in many systems, in the fly, in the vertebrate spinal cord, and indeed in our hands, they bind also. So this is simply an IP where we've pulled down with LHX2 and we probe with LHX2 and LDB1. And in our hands, in our system, in the dorsal telencephalon of the mouse, these two factors bind. Okay. Now, they bind each other and they work together, but they don't regulate each other's expression. So here is an LDB1 cortex-specific mutant using EMX1 Cre. And in the LDB1 mutant, you can see no perceptible change in LHX2 expression. LDB1 does not prevent LHX2 from being expressed. It's present, but the loss of LDB1 indeed prevents LHX2 from functioning. And that's what I'm going to show you now. So here is a close-up of the medial telencephalon of a 12.5 mouse showing the marker FB1. And you can see that FB1 excludes the hem and then is very intense right where the hippocampal primordium begins and then tapers off. That's control. Now, in the LHX2 cortex-specific mutant and in the LDB1 cortex-specific mutant, FB1 expression is reduced to a mere shrunken patch because the hippocampus in both these mutants, cortex-specific mutants, is known to be an incredibly reduced shrunken structure. Okay. What remains of it may be because of hippocampal cells that were formed before the cortex-specific, the EMX3 kicked in. Okay. Is this lack of hippocampus because something happened to the hem? EMX1 Cre doesn't actually act in the hem, and nothing seems to have affected the hem from forming. Okay, it's hard to talk about extent of hem and so on. Yeah, it looks a little bit bigger here. But the telencephalons of both these are shrunken enough that I just wanted to show you that the hem is present, or at least Wnt genes are present in the medial telencephalon. They're supposed to induce the hippocampus, but for some reason they haven't done so here. And I'm going to explore a little bit why. This, I should say, was the thesis work of a really gutsy student, Veena Kinare, who has left the lab now and is in industry. And a postdoc, Archana Iyer, who's taken on this project and is taking it into further directions because it opened up something really cool. Okay. This just came out a month ago, so it's available. Okay, why is LDB so interesting? LDB is interesting because of this dimerization domain. So here are two LDB molecules, and they self-dimerize. And their other ends, okay, this limb interaction domain here permits this LDB molecule to interact with molecules that have limb domains, such as the LHXs. So here is an LDB interacting with the limb domains of a LHX molecule, which also has a homeodomain, which binds DNA. And because of this dimerization ability, LDB is able to adapt, serve as an adapter to multi-protein complexes and effectively loop chromatin, so bring distant domains together. Okay, LDB has been shown to interact with all of these factors, Carter factor, VHLHs, limb-only genes, LHX, OTX, and so on. Now, why this is extremely interesting to us, I'm getting a little bit ahead of myself here, but we have data with LHX2 occupancy, ChIP-seq, showing that broadly LHX2 doesn't bind near the transcription start site of its target. It binds far, far away. And this LDB-mediated looping may be what actually brings the whole complex together to regulate the target genes. Now, this looping model has been beautifully proposed, but the interaction of LHX and LDB has been exceptionally well studied in Drosophila, where this tetrameric complex was proposed. So this is sort of essentially just this, where the DNA has just been made shorter for ease of representation. So here is an LHX LDB tetramer, where the LDB dimerizes and each LDB molecule binds an LHX gene. So this model was actually proposed for Apteris and CHIP in Drosophila by Steve Cohen's lab and John Thomas's lab. And they showed that this tetramer is the active complex by simply looking at how the stoichiometry works and by how this tetramer can be poisoned. Okay, essentially, so they introduced truncated versions of these. A limb-only molecule introduced would poison this tetramer, or a truncated version of the LDB of the chip would poison the tetramer. And their assay was the fly wing, where Apteris function is critically needed to specify dorsal fate in the fly wing. So it was based on these studies that we modeled the experiments I'm going to talk to you about. What was exceptionally cool about these studies was that they created a chimeric construct. Okay, they essentially cut out the middleman, cut out these binding domains, and created a construct which essentially has the homeodomain component of the Apteris LHX and the dimerization domain of the LDB chip. Okay, so this chimeric construct essentially self-dimerizes, and if the tetrameric model is right, then this chimeric construct would do the job. And indeed, in these fantastic papers, it did. It was able to rescue the poisoned tetramer and the mutants of Apteris and CHIP and so on. So we decided, hey, okay, so if it worked in the fly, and if this mechanism is evolutionarily conserved, then maybe it would work in the mouse as well. And Hari Padmanabhan, a former PhD student who has now finished his postdoc with Jeff Macklis and is now working in industry, made this construct as a sort of parting gift to the lab. And Veena and Geeta and Archana took it on. And what I have to show you is essentially how this construct can rescue hippocampal fate in the LDB1 mutant. Okay, so here's what they did. Geeta was able to electroporate 10.5 embryos very precisely medially, and control GFP or construct with GFP. This is the chimeric construct introduced in the LDB, in the cortex-specific LDB mutant. Okay, so remember the LDB mutant has LHX2 expression. Okay, there's no problem with that, but that LHX2 cannot dimerize because there's no LDB. And that's why introducing this construct, which has the self-dimerizing ability, we hypothesized could change things. So here our control, FP1 and LHX9, just another marker for the hippocampal primordium. And here is the non-electroporated hemisphere of the cortex-specific LDB1 mutant, where you can see that there is very little hippocampal neuroepithelial expression. Here is the electroporated hemisphere of the same brain. And you can see that there are now additional patches of FP1 which line up with the green electroporated clumps, and additional patches of LHX9 which line up with the electroporated clumps, and here's a false color overlay. This is electroporated at 10.5 and we looked at 12.5, and we were heartened to see that it looked like some type of hippocampal specification might be happening here. So we pushed it a little later, and here Geeta electroporated at E13 to E14. And this was sort of, we didn't expect this to work, because by E13, 14, it's late in the day, the hippocampus is specified, right? Well, turns out that those cells are still, sort of willing to process instruction if they receive it. Turns out, looking at P0, here is neuro D in the control, nicely expressed in the dentate and a bit of CA3, the non-electroporated hemisphere, a very shrunken hippocampus, barely expressing. Two other markers, this is ZBTB and this is ZBTB20, the entire hippocampal primordium, and KA1 in field CA3. And now this is, and in the non-electroporated hemisphere, barely expression visible. And now the electroporated hemisphere. Okay, yes, it's very tiny, okay, but this very tiny expression gave us great joy, and I'll tell you why. Okay, here ZBTB expression came up in a wider extent, and KA1 expression in a sort of intermediate extent. This gave us great joy because, okay, look how there is GFP revealing where the construct got electroporated all over the primordium. There are clumps and clumps of GFP, but only the bit that was sort of medial most, nearest the normal location of the dentate in CA3, only that bit expressed the marker. Okay, it's not like this construct just rampantly induced neuro D expression everywhere. And this regional readout continues with these other markers as well. Okay, ZBTB, for example, it's expressed all over the entire primordium. I'm sorry, the name is not there, ZBTB20. It's expressed all over the primordium, and look at these clusters that are farthest out from the medial edge, so sort of towards the subiculum, and indeed they express ZBTB20. Whereas Ka1, it's only a field CA3 marker, and this extreme clump that's right near the subicular end of the hippocampus does not express Ka1. So what I'm trying to say is there are two very striking things about this. One, that we were able to restore hippocampal marker expression in an embryo by electroporating it pretty late, at E13 to E14, and looking at B0. And two, that this construct, which is essentially LHX2 homeodomain that was given the ability to dimerize by its chimeric nature with the LTP, this construct, it didn't rampantly just induce hippocampal fate. It basically gave a regional readout where it gave field-appropriate identities, suggesting to us that the job of this construct, and by extension of LHX2 endogenously, may actually be to allow a responding cell to read out an instructive signal and respond in a regionally appropriate manner. Okay, so not to induce identity itself, but to be part of the response machinery, which is an interesting and very complex job, and entirely fitting with the hypothesis that Wnt signals from the hem in a graded manner induce distinct hippocampal fates. Okay, so does LHX2 then help presumptive hippocampal cells to process the Wnt signal? This is the key question. Okay, I don't have the complete answer for you, but I have a little tantalizing nugget. Okay, so this is LHX2 expression. It's not at the hem. And here is LEF1 expression in the control. LEF1 is part of the transcriptional complex that processes canonical Wnt signaling. So we're broadly looking at canonical Wnt signaling because it's well established. Actually, in a collaboration I did as a postdoc with, when I was a postdoc with Liz Grove, a collaboration with Andy McMahon, we looked at the Wnt3A knockout. Wnt3A, the earliest Wnt gene expressed in the hem, that knockout completely lacks the hippocampus. So we were pretty sure that it was Wnt3A or canonical mediated signaling that induced the hippocampus. And LEF1 is one of the canonical Wnt transcriptional complex molecules, LEFs, TCFs. And I'll say more about this in just a little bit, but I'm just trying to show you this. Okay, this is LEF1, very, very intense in the bit that's right next to the hem where the dentate would form. And then it tapers. Okay, the protein gradient, I don't show it here, but it looks exactly the same, intense here and tapering. Now look at what happens to LEF1 in the cortex-specific LHX2 mutant. Okay, these are 3 different embryos. This is the work of Mallika Chatterjee, a former postdoc in the lab. 3 different embryos. There's 2 things that jump out at me about this. Okay, first, of course, okay, this great big neocortex, which doesn't appear to have LEF1 expression, now has it. Okay, has it at somewhat low levels. I don't know what it does there, but what's most interesting to me is that this giant patch of tissue here, all of this is not hem. Okay, this is distinctly a bit is hem and the rest is hippocampal primordium. That intense patch of LEF1 that should be right where the dentate CA3 form is missing. And in fact, LEF1 here is completely gone. And this may be the relevant function of LHX2 because a study 2 decades ago, Galceron et al. From the Rothschild lab showed that the LEF1 null mutant is specifically missing the dentate. Okay, very cool finding that. So LEF1 is supposed to be a key mediator of canonical Wnt signaling, but it's not the only one. There are TCFs as well. And they made the very nice analysis in this study that LEF1 itself appears to be very critical for the dentate because you don't have it. It's also important for the rest of the hippocampus, the rest of the Ammon's horn, which shows distinct abnormalities. But they made another mouse with a LEF1 lac Z construct, which not only took out the DNA binding capacity of LEF1, but also created a dominant negative effect. And in this situation, it was able to essentially poison canonical Wnt signaling by binding potential sites, binding partner sites. And in this mouse, okay, the hippocampus was basically gone. Okay, so LEF1 by itself seems to be critical for the dentate and has some function in the rest of the Ammon's horn, but canonical Wnt signaling appears to be critical for the entire Ammon's horn. And now put this together with the fact that when LHX2 is removed in the cortex-specific mutant, what we seem to lose is this very intense patch of LEF1, and we seem to sort of extend this lower level of LEF1 through the neocortex. I don't know which of them is actually the most significant, but it appears to me that this particular function may well be. And this is what we decided to sort of ask molecularly. We wanted to then ask, okay, in situs and mutants are all very well, but does LHX2 actually regulate the Wnt response? And to do this, we had to extend in a direction that was at the time new for my lab. Okay, just to remind you that this is the normal LEF1, this is the cortex-specific LHX2 mutant. And for this, I have to thank an extremely talented former postdoc, Bhavna Muralidharan, who has just started her own lab at INSTEM Bangalore. She was funded by the Wellcome DBT, not only for her postdoc, but also as her, she has a junior faculty intermediate career grant from them. And she decided that, unless we look at chromatin occupancy and unless we analyze what's happening at the level of the chromatin, we're not going to understand this mechanism. So we collaborated in a very welcome and wonderful collaboration with Sanjeev Galande's lab at IISER Pune. I'd like to acknowledge his student, Saurabh Pradhan, and his postdoc, Molly. And I'd also like to acknowledge and thank a very young member of the team, my current PhD student, Varun, who actually trained in bioinformatic analysis with Sanjeev Galande. We're very grateful. And is basically, what Bhavna started, Varun is now continuing together with this collaboration. And we are now trying to analyze what is happening at the level of the chromatin. I have one slide of tantalizing data. We're still analyzing these vast data sets to try and figure out what this all means. But I'm just going to show you one little glimpse to just show you where we are headed with this project. Okay, so this is the LEF1 locus, a cartoon of it. And what Bhavna did was, given that, she based her whole study on the fact that we found that LHX2 has different functions in the medial and the lateral telencephalon. So there's no point in taking out the whole telencephalon and grinding it up and extracting chromatin, because we may actually have different functions in the two different places. So always embryos were dissected, and we're talking about hundreds of embryos over the different ends for each ChIP-seq. The hippocampus and the neocortical primordium separate. And here is an attack-seq profile of just accessibility. And see how right at the LEF1 start site and a little beyond, and of course at other loci still, we have basically where is the chromatin open. And this is just wild type. We have histone marks, the activatory H3K27 acetylation mark. We have the K4 trimethyl mark. We have the K4 monomethyl mark, which is a sort of putative enhancer activatory mark. And you can see it occupies in other regions. I'm just showing this to you to give you a sense of what's happening at the LEF1 locus and around it. Because when we look at LHX2 occupancy itself, okay, we get a different readout for hippocampus and cortex. Okay, what we get when the software calls the peaks is that this one peak, which is sky high compared to all the others, this is the one peak far distal from the transcription start site in the hippocampal primordial that LHX2 seems to occupy. And we don't get it called in the cortex. Okay, now remember what I said about LHX2 binding distal sites, not right near the enhancer, and the whole chromatin looping idea. So it's all maybe part of this story where LHX2 binding distally may actually be key to regulating LEF1 expression in the hippocampal primordium. Okay, so this is where we are at right now on this project. Of course, what we are dying to do is to analyze the data set that we already have as to whether all of these profiles change in the LHX2 mutant, to ask what is happening at the level of the chromatin for LHX2 to execute such an apparently dramatic effect between cortex and hippocampus. Okay, so just to summarize so far, I showed you that 3 fundamental transcription factors interact to position the hem, and that an LHX2-LDB1 partnership seems to be sufficient to specify hippocampal fate, okay? And it does so in a regionally appropriate way. And because of that, we conclude that LHX2 may regulate the canonical Wnt response within the neuroepithelium adjacent to the hem to allow it to acquire distinct hippocampal fates. Okay. So if canonical Wnt signaling is so critical to induce hippocampal fate, okay, here is the hem expressing Wnt3A, and here's the hippocampal primordium. But you know what? Why doesn't the hippocampus form on the other side? There is Wnt signaling, and this structure, which is the choroid plexus epithelium, wasn't always the choroid plexus epithelium back at 9.5 before it even formed. So what happens in the other direction? So I'm now going to sort of share with you some unpublished data in the whole area of what, if anything, canonical Wnt signaling does in the choroid plexus. So let me just first show you some expression studies, because we first wanted to ask, well, you know, is canonical Wnt signaling relevant for the choroid plexus? It's been shown very nicely by Jean Hebert's lab 2 decades ago that BMP signaling from the cortical hem is essential for choroid plexus development. The choroid plexus requires BMP signaling. A recent study from Helen Cooper's lab at Queensland Brain Institute in Brisbane showed that Wnt5A, non-canonical Wnt signaling, is important for choroid plexus development, but the canonical Wnt's have not been explored. And here then is the cartoon, and here is evidence that beta-catenin itself, shown in red, and LEF1, indeed, that's the hippocampal primordium, and that's the choroid plexus. Frizzled one of the many Wnt receptors, is expressed in the choroid plexus. Here is an immuno showing TTR, which is a transthyretin. It's a dominant secreted protein that's made by the choroid plexus and present in the CSF. And here's frizzled one, okay, at the apical boundary. Axin 2, which is a readout for canonical Wnt signaling. And here is the reagent we're going to use. This is LMX1A expression. And here is an LMX1A Cree mouse with an AI9 reporter to show where it's expressed. This we obtained as a kind gift from Kathy Millen at Seattle Children's Hospital. The cortical hem produces, sort of gives rise to its 2 lineage derivatives, one of them being the choroid plexus epithelium and the other being Cajal-Retzius cells, both of which are nicely red in this picture. So in all of the perturbations I'm going to show you now, we're going to use LMX1A Cree. And in a lovely collaboration with some new colleagues, the internet is a wonderful thing. You can not meet people and collaborate with them. Natasha Jovanov-Milosevic and her team in Croatia, we were able to examine a gestational week 11 very early fetal human brain. So we discussed with Natasha, and her lab did these beautiful immunos to show, so those are low mags and high mag, to show that indeed frizzled one and beta-catenin and aquaporin, this is the sort of water channel that the choroid plexus needs to produce cerebrospinal fluid, are all well expressed in the fetal human choroid plexus. So what I'm going to show you now is data that demonstrate that canonical Wnt signaling is critical for the choroid plexus, and that it regulates the choroid plexus bidirectionally, in that, you know, loss and gain both affect the choroid plexus, and that a precise amount of it seems to be necessary. And I'm going to show you this not only in the embryonic mouse, but also in human embryonic stem cell derived organoids, and that's where the human connection comes in. Okay, so everybody now jump out of the hem in this direction into the choroid plexus and not the hippocampus. Okay, take a right turn, not left. Right. So what is the strategy? The strategy for either increasing or decreasing canonical Wnt signaling is essentially a beta-catenin loss of function or gain of function. Okay, the beta-catenin loss of function mouse, this has been a well-published reagent, Anjan Chen's lab, where the Several exons of beta-catenin are just flocks and they get recombined. And remember, we're using LMX1A3 throughout. The gain of function is interesting. Exon 3 is recombined. And what this does is it removes phosphorylation domains where GSK3 beta would normally phosphorylate beta-catenin and tag it for degradation. If beta-catenin is not phosphorylated and not tagged for degradation, it essentially becomes constitutively active. So you basically have a gain of beta-catenin. Okay. The people who worked on this project, Mallika Chatterjee started this project. She's now a faculty member at Amity University in Delhi. Arpan Paricha, a new PhD student, and Varun Suresh, whom you met before. And here's the data. So beta-catenin loss affects the choroid plexus pretty dramatically. Okay, so here is TTR, and you can already see that there is something really wrong with the morphology, the assembly of the choroid plexus. Ecadherin is messed up, and OTX2 shows that there are large tracts of the choroid plexus that don't even have this essential transcription factor, which is necessary for choroid plexus development. And aquaporin, okay, which is the major channel that by which the choroid plexus can secrete fluid into the cerebrospinal fluid. Again, aquaporin seems to be missing in large tracts of the choroid plexus. This demonstrates then that canonical Wnt signaling is necessary and important for choroid plexus development. Now, what if you have too much of it? Okay, so when we looked at the gain of function mouse in a similar situation, this gave us some surprises. So here you see in the gain of function mouse, the choroid plexus seems to be sort of losing its territory. Okay, so axon 2 shows that there is certainly gain of function of beta-catenin because axon 2 is a target. But see how PROX1, okay, which normally labels a dentate and a bit of the hippocampus, see how neurog2, which is normally only in the neuroepithelial ventricular zone, and neither of them are seen in the choroid plexus in the control. See how they've now begun to come up in the choroid plexus, okay, very distinctly so. And in fact, what we found is that gain of beta-catenin function turns the choroid plexus into a hippocampus-like identity. Okay, so we found this by dissecting out the choroid plexus and the adjacent hippocampus. So from an embryonic 14 mouse, this was challenging, but not as challenging to identify what's what, because we simply use the LMX1A3 AI9 background. So the LMX1A3 both remove the beta-catenin exon 3 and the stop flocks reporter, so we could see which tissue was red when we dissected it out. And here's a simple PCA analysis, which shows that the control choroid plexus is over here, and the mutant choroid plexus is way over here, in some sense closer to the control and mutant hippocampus. Okay, this is borne out when we look at the genes themselves. So here you have 2 replicates of the control choroid plexus, 2 replicates of the GOF, the gain of function choroid plexus, and 2 replicates of the control hippocampus. And you can basically see that the choroid enriched genes and the hippocampus enriched genes, okay, you can see how the control choroid plexus and the control hippocampus look very different from each other, but the GOF choroid plexus is basically now looking more and more like the hippocampus. It's lost the choroid enriched genes and it's gained the hippocampus enriched genes. Okay, these are sort of characteristic genes that are necessary for choroid plexus function. So at the top you see aquaporin, a whole bunch of channels, sodium and potassium channels, other solute transporters. They're all broadly turned off or decreased in expression. And this is important because this explains why in our gain of function mice, we typically saw collapsed ventricles. Okay, we basically saw very little cerebrospinal fluid. There isn't enough cerebrospinal fluid in these mice because all of the transporters seem to be affected. And the reason this is important and the reason why we wanted to look at, so gain of function is normally thought of as a abnormal thing, you know, why would one even study it? Loss of function is sort of more looking at sort of what the gene does in vivo. Well, it turns out that human mutations in beta-catenin have been reported to have the same stabilizing effect of beta-catenin. So basically, the beta-catenin human mutations, GOF mutations, are reported, and they have broadly been studied in their role in carcinomas. Choroid plexus carcinomas have been studied, but not in their role in affecting the cerebrospinal fluid. And we think that this is a new territory to explore, because it turns out that gain of Wnt signaling, canonical Wnt in the choroid plexus, seems to dramatically affect the ability of the choroid plexus to secrete at all, and certainly to sort of the elements of what it secretes. So here then is our foray into human embryonic stem cell organoids. And for this, I would like to acknowledge our wonderful collaborators, Orly Reiner's lab at the Weizmann, her student Aditya Kshirsagar, and Lihi Sriya, and Arpan, who Orly's lab kindly hosted for the better part of a year. He basically went there and learned how to make organoids and is now setting up the technology back home. To do this, he used Sasai's well-published protocol, where they essentially, at day 18 in the stage after aggregation and transferring and so on, they induce with morphogens. And these morphogens have a specific well-defined configuration of the MP4 and KIR, which is a GSK3 beta inhibitor. So it's essentially a canonical Wnt agonist. It does the same thing again, stabilizes beta-catenin. So treatment with this morphogen essentially produced choroid plexus-like structures. Okay, so here is just images of how the organoids grow. Day 18 is when the induction happens. And you can see these blebs of these organoids in their peripheries begin to express TTR. Okay, not only do they express TTR, they express OTX2, and they express, so this is the organoid, and this is a mouse choroid plexus, and you can see how they sort of look very similar. So it looks like we have choroid plexus-like fate. Here's an example of 3 of them. And what's interesting is that instead of adding the low dose of the KIR that is necessary for choroid fate induction, if we overdo it, if we create a GOF-like situation, we lose the choroid plexus identity. Okay, so here are just 3 examples, and here is qPCR, where day 18 the blue bars are, not they're taken as 1, and basically by day 18, with the if at day 18 the 3 micromolar KIR is added, then we get choroid markers induced. But if we add too much of it, if we add 12 micromolar, we basically lose these markers. Okay, and LEF1 shows a steady induction consistent with a low level of canonical Wnt activation and a high level of it. Okay, here is just the pictures. You can see TTR, aquaporin, and OTX2. You don't even need 12 micromolar KIR for it. Basically, 6 or 9 micromolar will do the same thing. It just removes the signatures of choroid plexus. And now I'm going to show you a high level of OTX2. This is a little interesting, because we get ZBTB expression, okay, by immuno in some of these organoids. Okay, it's just one marker. It's not hippocampus. It's just one hippocampal marker, but it's tantalizingly parallel to what we see in the mouse. Okay, so here's just a quantitation that the number of OTX2 positive cells is very high in the 3 micromolar and then down in the rest, and the number of ZBTBs increase. But here is, so this is just a normal 3 micromolar induction where choroid plexus markers come up by day 30. And here comparison of 3 and 12 micromolar, low and high KIR. Okay, what has come up has gone down again. So essentially, in the choroid plexus, sort of like organoids that we've created, high beta-catenin activation seems to take away choroid plexus fate, broadly allowing us to conclude that in this structure, which develops on the other side of the cortical hem, we need a controlled balance of Wnt signaling. So to summarize, that's just a cartoon summarizing it, Wnt loss in GOF and in the organoids. I'll end with just 2 slides of sort of where this cortical hem fits into a broad picture of telencephalic patterning. Because until now, I've stayed in the medial telencephalon and hippocampus and choroid plexus. There is a broader story, and this was explored by my former student, Achira, who is now finishing up her postdoc with Kathy Millen at Seattle Children's. Achira noticed that, okay, loss of LHX2 does expand the hem, but it doesn't expand the hem very, very rostrally, where there isn't any hem to expand. Very rostrally, there is a bit of non-hem tissue in this mutant. So she began looking at it, and to summarize the data in one slide, what she essentially found is that the hem is part of a tripartite forebrain midline sort of signaling system. Okay, up rostrally, here you can see it better in this sagittal view, where the septum ends, the hem begins, and where the hem ends, that's where the thalamic eminence begins. Eminentia thalami, maybe you can see it better in this section. So the choroid plexus transitions into the eminentia thalami, which is the connection of the telencephalon with the diencephalon. It's sort of like the hem of the diencephalon when it connects to the telencephalon. What Achira found is that rostrally, in an LHX2 null mutant, basically the septum expands and takes over what little bit of non-hem structure could have been cortex. There isn't any cortex even there, it's septum. And caudally, the eminentia thalami takes over and basically expands. So this then, oh, and I'd like to acknowledge that this was a lovely collaboration with Gundela Mayer and Alessandra Pirani. It was such fun interpreting all of these phenotypes together. So what we see the hem as now is part of a forebrain hem system. Okay, this is a horizontal section of mouse, and I've put a horizontal section just because it has a very nice morphological parallel with this horizontal fetal human section. Okay, this is 6 gestation weeks from Gundela's collection. This is labeled with calretinin, and here we've used LHX5 just because it labels the similar structures. You can see the hem, and you can see here the transition of the caudal hem to the thalamic eminence in both species. The point is that the septum and the thalamic eminence predated the appearance of the hem. Okay, in evolution, fish and frogs have septum and thalamic eminence, they don't have hem. And with increasing complexity of the brain, this forebrain hem system seems to have added a new element, the cortical hem, so that perhaps the telencephalon may form, may expand, hippocampus may be induced. So to just place this in context, while we are studying this one signaling center, we should really see it as part of a whole signaling system that patterns the entire telencephalon. Okay, I think I'm probably out of time. How to make a hippocampus was how we started this talk, and indeed how I started my lab. And answers we have so far seem to be, you make it by making an organizing center and a responding tissue. And the control of that seems to be LHX2, where LHX2 isn't is the organizing center, where LHX2 is the responding tissue. Then you restrict this to the correct location. And the way you do it is in collaboration with PAX6 and FOXG1. Now, remember, we don't actually have the answer as to why all of this action happens right in the medial telencephalon. We don't know why LHX2 turns off only in the middle, right in the middle of the telencephalon. Potentially, that happens when the neural plate is open, and what's going to become the middle is actually outlateral, and there are potentially interactions with non-neuroectoderm tissue that tell the medial telencephalon the medial edge is going to be the medial edge long before it ever came into the middle. So we don't have that fundamental answer, but this sort of opens the door to it. Okay? Then you regulate the responsiveness, okay, of this organizer in the adjacent tissue. And this adjacent tissue has to be regulated on both sides. Okay, what I showed you was how the LHX2 LDB1 complex acts to specify hippocampal fate in the absence of LDB1 in the hippocampus, and how beta-catenin-mediated Wnt signaling is critical for the choroid plexus in a regulated manner, not too much, not too little, in order for the choroid plexus to be itself. Okay, and then this organizer is part of an evolutionarily conserved system of organizers so that the brain may be controlled, developed, and may evolve. So that, friends, is our story of how you make a hippocampus. Would that you could do it like this and plant bright ideas when you wanted to. But I think this molecular complexity is more interesting. And I will stop and thank you and take questions.
So thank you very, very much. You can unshare your screen, especially for this fantastic, story about how actually the brain is orchestrated. For me, it's this Wnt signaling, especially, has always been so fascinating because it seems that can do a lot of things during development, not just patterning these sites, but so many areas can be patterned. So it's very fascinating to see how much all these tiny changes during development can make so many other changes. So one thing, yes, you should, yes, leave your.
Sorry, I'm trying to figure out how to unshare the screen.
And then maybe I can start. I have quite some questions, both on the human and mouse side. Maybe yes, the first and more obvious. It's about how we can then make all these RNA organoid. Obviously, it's a difficult thing to do. You're still not sharing, yeah?
Yeah, sorry, I'm just
have to look and click in your face.
There we go. Okay. All right.
So one thing that I think is would be very, very beautiful is to manage to make a pure real hem and anti-hem in the organoids, right? So
yes,
that could be something fantastic, and I think there is the potential because by maybe modulating and playing with all these different genes that you have now in the mouse, the possibility of making some regionalization there and some patterning would be fantastic. I mean, I just have in mind the beautiful paper from Lawrence Studer lab, where they actually make the signaling center to make the dorsoventral regionalization, and so this would be fantastic. So what do you think?
So actually, Sasai's lab in 2015, Sakaguchi et al., the reference, they actually did produce very beautiful hippocampal likes of OXG1 expressing and hem and choroid plexus all in the same organoid. It was gorgeous.
And do you think that it's possible to have also an anti-M?
Yeah.
Why not?
Why not? Why not? I mean, so the organoids, depending on, I guess, how big and how complex they get, they may not all be one thing. So I think you look at them very carefully. And since all of these were done actually in Orly's lab, I saw the sort of pictures. I didn't get to actually put my eyeballs on it. So my sense is, I mean, you would know this. My sense is that if you create the thing big enough, you have to, treat it. I mean, Sasai's protocol says how they divided it in half and kept it in high oxygen and so on, because otherwise maintaining it without fragmenting was difficult.
But I was thinking also by forcing the suppression or the downregulation of some genes, like checks or PAC6 in specific position. I don't know if you recreate something that is similar to what you observe in the mouse.
Create high PAC6 potentially.
Yes, or maybe using something like to modulate the expression in specific time windows or something like that. That would be fantastic.
Yeah. So, well, and then the antihem is the source for the amygdala, the paleal amygdala. So then the next question would be, does it produce amygdala?
Yeah, exactly. And also the problem is that you have to somehow give a specific time window in which you can give this additional patterning, right? Because with the organ, it's a bit more difficult because you don't have, you can have choroid plexus and you can also generate some CSF, but it's probably not so well regulated like in vivo.
Yeah. I mean, actually, organoids, yes, but human patients exist with these mutations, right? Their CSF has not been analyzed, to my knowledge.
In organoids?
No. Patient CSF. Because Wnt gain of function mutations have been characterized in humans, right? They cause cancers, they cause carcinomas of different kinds. So this
gain of function mutation that you mentioned before, also they seems to be incredible that they exist actually, because considering all the function that Wnt signaling has in brain development, the fact that there are actually some very simple phenotypes, like in which you are, for example, the collapsing of the ventricles or something like that, is quite surprising, right?
Well, I mean, so, it's probably not the whole choroid plexus that is gain of function in the human, right? I mean, the mutation may happen in sort of small clones, or sort of not, if it was the entire choroid plexus, we would have other problems then. But yeah.
So maybe I can ask you the next question if someone else in the meantime is curious about one of the other aspects. So is about the mouse work. The mouse work, I was also wondering one thing. So all these patterning factors that you described so wonderfully, they have this function during generation of different area, but they also seem to have sometimes function in the generation of different cell types. So could you comment if you have different cell type of regulation of differentiation? So when, for example, you got this multiple hippocampus, for example, did you get all the cell type inside and how they develop and how they were when they were adult, if you have the chance to look at them?
Yeah, well, so unfortunately they died at embryonic 17 because they died with pale livers. So LHX2 is critical for erythropoiesis, and we think that maybe there is a non-cell autonomous effect in the liver, because the chimeras didn't have patchy livers; they just had pale livers. So basically, they didn't make it past E17. Otherwise, we would have done experiments on mice running around with extra hippocampi. But Nidhi is trying that now that we know that FOXT1 and PAX6 expression sort of restrains the hem from forming laterally and so on. We're going to use a triple mutant and electroporate Cre and create a lateral hem and ask how does it make a lateral hippocampus. So we're on it, but it's one of those dream experiments.
So when you have also this very nice choroid plexus phenotype in the last story that you presented in the mouse, so this different gain of function and loss of function in the choroid plexus. So do you also I'm very curious about the anti-M. Did you also have changes in the other side?
In the mouse?
Yes.
So PAX6 is necessary for anti-hem fate. And in the absence of PAX6 by itself, the anti-hem is not detected. Mm-hmm. To also remove LHX2, so something happens at that lateral boundary and you push the anti-hem down even further. So we reported it and we didn't sort of study it that well, but there is some interesting action going on there with cross-regulation with the GSH2 and
exactly. So
there is some unexpected action at the anti-hem in those mutants. So it's not like we just got a combined effect, but the hem did extend all the way.
So did you ever look at all in your organoids, all these genes expression? So in human, all the gene expression, so that you have been showing in a mouse?
So, sorry?
The gene expression, the LHX and LDB1, for example, in the organoids?
Oh, no, we didn't look at LHX. Yeah, but if Arpan is in this talk, my student, that should be on your list. I mean, it's probably there. It's probably there.
Yeah, no, I can imagine. But if there is in any way any distribution that is different between them.
Yeah. No, so we should characterize. You're pushing me to sort of we should characterize the regionality of the organoids better. Right now we were just so happy to have in the periphery the choroid plexus markers. So we're pushing to try and get hippocampal markers there. But that's a bit of a reach to expect every single thing to.
No, of course, and they are much less organized. So of course, but it would be fun.
Yeah. But the middle of the organoid is a little vast uncharted territory. So what you're suggesting is that we look at it and chart it a little bit. So yes, no information yet on our list.
Yeah. Good. So Shubha, thank you very much. If there are no more questions, then
thank you everybody for coming and.
Thank you very much for this fantastic talk and really for this great overview of this part of development, which is the patterning part, which is all the orchestration of this development of the brain. So thank you very much again and have a good night.
Thank you. Pleasure. Bye, everyone.
Bye, everyone.